Good evening. I'm the Biological Secretary of the Royal Society, and it's my pleasure to welcome you to this year's Croonian lecture, which will be supported by a British Sign Language interpreter and by live speech to text. The Croonian lecture is the Society's premier award in the biological sciences, and is named for William Croon, one of the original fellows of the Society. The series began in 1738, when his wife bequeathed 1/5 of the rent of the King's Head Tavern at the corner of Lambeth Hill and Old Fish Street to support it. This year's lecturer, Jonathan Ashmore, is Bernard Katz Professor of Biophysics in Neurosciences, Physiology and Pharmacology at University College London. He is renowned as a child film star and for communicating neuroscience to older people of my generation by exciting nerve cells using their favorite rock and roll music. But tonight, we are recognizing his contributions to our understanding of the mechanisms of hearing, in particular for his work using a combination of biophysical, optical, and electrophysiological techniques to describe the novel electromechanical motor of the mammalian inner ear. His lecture tonight, as you can see, is on the neuroscience of deafness, and we look forward to it very much. Jonathan.
Thank you very much. Well, thank you very much indeed, John. It's a great pleasure to be sitting here, standing here, rather talking to you. And I thought we'd begin just with a picture of another gathering in our academy. This is an academy of the Royal Academy of Arts. It's a picture painted by John Zoffany in 1771, and you can find it up the road in the Royal Collection, in the Queen's collection. But in the centre of this picture here, there you will see is the President of the Royal Academy of Arts, Sir Joshua Reynolds, and if we zoom in on that, you will see that, in fact, there's actually something else which is centre stage, which is the ear trumpet which Reynolds used to wear. He became deaf in his 40s, but wearing using an ear trumpet didn't, in fact, affect his sociability or his social life at all, one gathers, at least from the description of Fanny Burney. So I want to tell you today about a particular class of cell, which John alluded to, the outer hair cell. They're called hair cells because they have projecting on their surface hairs. In fact, there's absolutely no connection whatsoever with the hairs on your head. And on the same scale, a human hair would be about that big long, so you could fit several of these into the diameter just of one human hair. But they're called hairs because when the light microscopes became powerful enough to be able to see some of these fine structures, they resembled hairs to the people who were actually looking at them. But they're also cells which fulfill a remarkable function within the cochlea. They're the cells which essentially provide the biological hearing aid which you have within your ear and which helps you to hear. As well as the hairs, they're cells which sit in a sheet of cells. I'll show you some pictures in a moment. They have a cell nucleus, and this end here communicates and receives information from the brain. So they form a part of an amazingly complex little bit of machinery, which is really the way in which the brain discovers the sounds that are surrounding us the whole time. They allow us to hear speech, they allow us to hear where a sound comes from, and they tell us whether or not to pay any attention to those sounds when they do actually appear. So it's a huge honor to be standing here in front of you. as a Croonian lecturer. And astonishingly, it seems that I'm actually the first sensory neuroscientist to be delivering this Croonian lecture for nearly 86 years. Edgar Adrian delivered a Croonian lecture on the messages in sensory nerve fibers and their interpretation in 1931. And he said something which is very prescient and still actually applies even today, which is, he says that the nervous apparatus which intervenes between stimulus and sensation is a dangerous topic, since it forces us to consider the mind as well as the body, and to attempt the measurement of phenomena which lie outside the framework of the physical sciences. So what I'm going to be telling you about this evening is really the machinery that is susceptible to the physical sciences. But it's a complex bit of machinery which does the hearing in your, for you. And then the process of actually listening, which is the brain's function, is something maybe that would be a topic for a future Croonian lecturer. Hopefully sometime before another 86 years have intervened. So let's begin with a little experiment. So what I'm going to play you is a sound, and I'd like you to indicate by raising your hand whether you can actually hear it. So there's a clutch of hands at the back there, and I'm assured both by my grandson and by the audiovisual people in the Royal Society that the quality of the sound is actually audible. That's a sound which is 16,000 hertz. Don't worry if you can't hear it. I can't hear it. But let's see if you can hear this one here. So this one here is a slightly different pitch. More people can hear that. Thank you very much. So that's 8,000 hertz. Let's try again. Oh, very loud. And let's go down to 2,000 hertz, another octave. Oh, that's that. And eventually we have 1,000 hertz. So you can see that depending who you are, you hear different frequencies of the sound. And one way of displaying that, of course, is when you go to an audiologist and have your hearing tested. They will test your hearing as a way, the amount of hearing you've lost relative to a normalized sort of human population. So when you're 20 years old, you have hearing which is probably pretty good for a 20-year-old population. This is the 16 kilohertz point, which many of us couldn't actually hear. But as you get older and older and older, your hearing, particularly at the high frequencies, begins to deteriorate rather significantly. I wear hearing aids, and although I would normally be sitting here, wearing hearing aids actually makes me audiologically 20 years younger. So I can't recommend them too highly. But the question is really, why do we start going deaf? And it'll turn out that, in fact, it's this population of outer hair cells which play a critical role in this particular function. Now, of course, it's not just age which is the risk factor for losing your hearing. There are many other things as well. For example, excessively loud sounds, the side effects of some drugs, and even some of the genetics, which we're beginning to understand now, can lead to various forms of deafness. So this is the more conventional way of showing the hearing range. It extends from about 60 or 50 hertz all the way up to about 20 kilohertz or so, at least when you're young. And this is a scale of a piano, and you can do a sort of DIY audiological test by seeing whether you can actually hear a tone at the top note of a piano keyboard, which is 4 kilohertz. If it just sounds like a plink, then you may have some sort of early mild or moderate hearing loss. Now, if you compare that with the hearing range of a mouse, which is the most favorite experimental animal that's used for studying many of the physiological aspects of hearing, a mouse's hearing starts at 1 kilohertz and then goes several octaves higher up than us, up to 64 kilohertz and even higher. So the hearing range is vastly extended, but the interesting thing is in terms of doublings of frequencies or octave ranges, it's not actually all that much difference from ours. What you saw when you came in was a spectrogram which showed the display of the sound being picked up by the microphones, spread out along the frequency range and then rolling through in time. And you can see when you see the word cochlea, it's got both high frequencies from the initial component of the C, and then the lia, which gradually builds up, and the A, which is essentially mainly sort of low frequency sort of sound. So how is it that you can hear these sounds? And the way in which you can hear these sounds is because in our ear we have a mechanical device which separates out the sounds according to their frequency. This structure separates out both low frequencies and high frequencies, and then there are nerve cells placed along that structure, which you'll find out is called the basilar membrane, which then send that information up to the brain. We have about 30,000 nerve fibers that communicate between our inner ear and our brain, and it's that information which the brain then computes meaningful information about the sound from. So this is what happens when you hear a sound. Here's the outer ear, and so the sound goes in through the ear canal, it vibrates the eardrum, and then the eardrum leads to a vibration of this pattern of little bones, the ossicles in your middle ear, and then eventually it gets transmitted to the cochlea of the inner ear. Now the cochlea itself is really just a cavity within the bone, a very sort of hard temporal bone that you can feel behind your ear. And so when we talk about the cochlea, when you see a picture like this, it looks as though it's actually sticking out. It isn't. It's buried in the bone, and that's made the study of the cochlea really very, very difficult, because, at least in the mammalian system, because it's been difficult to get at. If you do dissect it out, and this is done by one of my colleagues at the Ear Institute, you can see that the actual human cochlea is about the size of a pea, very small. And again, it's the small size which has made the progress on trying to understand how it works difficult. It's inaccessibility and its small size. It's called a cochlea, of course. Don't need to remind you, because that's the Latin for a snail. And so this spiral structure here is essentially the essential component of the inner ear, which leads to hearing. So let me now start telling you a little bit about the structure of the cochlea, how it works as a physical device, and then the role of these hair cells within the cochlea. So I'm sorry about that. Let me go back to where I was. Help. Nothing like giving away the secrets before you've even started. So let me see if I can get back to the beginning. Thank you very much. I don't want to run through the entire talk backwards. Much obliged to you. Thank you very much. So perhaps in the beginnings, the first person who really took an interest in how the cochlea might work was Hermann von Helmholtz. He was a polymath, foreign fellow of the Royal Society, and in 1864 he actually gave a Croonian lecture. Now, although at the time he was clearly thinking about how the ear worked, his Croonian lecture is actually about the eye movements. So we had to wait for a little bit longer for him to write the textbook where he elucidated his ideas of what the actual machinery was. What he did at the time was, because the anatomy of many structures was becoming much more apparent. Microscopes were getting better, material for fixing the soft tissues was getting better. People had begun to understand what detailed microanatomy looked like. And so Helmholtz had available to him a view of some of the finer structures of the cochlea. He thought the cochlea looked like a piano. In fact, he was a very keen musician, and to some extent that's actually correct. He thought it looked like a piano, but he couldn't do any experiments on it. What he could do, however, was devise mathematical models. And the model that Helmholtz devised really forms the basis of many of the ways in which we now think about the cochlea. It took another 70 years for the experimental verification of Helmholtz's ideas to become clearer, and this was done by the work of Georg von Beckerschi. And Beckerschi was awarded the Nobel Prize in 1961 for his discoveries, quote, the discoveries of the physical mechanism of stimulation within the cochlea. So he used a number of clever techniques to show that various structures within the cochlea were actually moving, and that was possibly the basis for separating out the frequencies of that were then sent off to the ear. So let's get down to business. Well, I think I can probably now reveal myself as more of a biologist than a physicist. There's this interesting situation that occurs within the Royal Society. You may be aware that physical scientists tend to wear ties, and biologists tend to not wear ties. So thank you very much, John, for allowing me permission to change my garb. The cochlea, if you uncoil it, is a tube, in our ear about 3.5 centimetres long. It's full of fluid, and this is in fact the role of the middle ear. It conveys sound moving in air to sound being transmitted in fluid. And as I'm sure you're aware, that's a difficult proposition, so you need a bit of essentially leverage to be able to ensure that the fluid movements are adequate. Down the middle of this tube, there's a structure known as the basilar membrane. And the basilar membrane is a dividing partition which is set up in vibration by the sound entering from the stapes, the middle ear, of the middle ear ossicles. Now, what is Helmholtz's model? Helmholtz saw in this basilar membrane a set of fibers. And he said, aha, it looks a little bit like a piano. And he knew that the treble strings in a piano are the ones that vibrate at high frequencies, and the bass strings are the ones that vibrate at low frequencies. So the kind of idea was that spotting the fact that there were fibers all the way along the basilar membrane, the high frequencies are the ones that it was posited were the high frequencies were the ones that produced excitation at this stapes end of the cochlea, and the low frequencies were the vibrations that were produced at the furthest distance away from the stapes. So there's a separation of frequencies all the way along the cochlea. But on top of that, the idea that the way in which a piano works is by saying, well, the treble springs are rather short and they're also quite stiff. And so by changing the stiffness of these fibers from one end to the other, you could end up with essentially an arrangement which separated out high frequencies from low frequencies. Well, this is an idea which is very common and well known to physicists. And I have here a little demonstration that's sort of known to all GCSE and certainly A-level students, which is if I have a spring with a weight at the end of it, if I vibrate the spring up and down, that weight will follow. And so if I plot the relative amplitude of the movement of that end, nothing very much happens at low frequencies. And likewise, if I vibrate it up and down at very high frequencies, again, nothing really very much happens to this weight. And so the movement again begins to fall off to zero. If I could move it fast enough, there'd be no movement at all. But there's a certain frequency with which I can vibrate that spring, where almost no movement at one end of the spring produces a large movement or resonance at the base. And so you can see that there's this particular frequency where you get a very large relative movement of this mass here. And that's exactly what happens in the resonant behavior of the strings in a piano. And the other feature of this is that it's only a really narrow range of frequencies which produce that resonance. Excellent. Now there's a snag, and the snag was known to Helmholtz, and it was also known to von Beckersche, and that is that if you try and do the same experiment, but putting this weight in some rather viscous jar of goo, it won't move very much. Now I thought it'd be a slightly messy experiment to do at the Royal Society, so I'm afraid I've flunked out of that one, but I hope you can imagine that that's actually the case. And so you can see this resonance completely disappears, and in fact the range of frequencies which are at all effective becomes much broader. And Helmholtz knew that you couldn't actually play a piano underwater. I don't know whether anybody's tried. I discovered the other day that there's actually an underwater festival in Florida. Apparently you have to get quite close to the musical instruments to be able to hear them. So that's the kind of the underlying physical idea. And to show you how this works in a model of the cochlea, we have to use one extra thing as well. So the question is, how do we deal with this problem of fluid viscosity? If there is fluid viscosity, it's going to damp out any resonance. And the answer was originally proposed by Thomas Gold in 1948. And he said there had to be some mechanism which fed back energy into the movement to essentially cancel the viscous drag that's going to be on those fibers of the basilar membrane. This is an idea which originally came from his work in the radar business during the Second World War. And he tried to demonstrate one of the consequences of that, which is that the prediction is that some of the energy which get fed back into the basilar membrane ought to leak out again out of your ears. But at the time, the microphones weren't sensitive enough to be able to detect that. And it then fell to a colleague of mine, David Kemp, at University College London, to show that with the new improved microphones that were available in the late 1970s, that you could measure sound coming out of the ear. And this is now used essentially as a technique to see whether newborn babies can hear sounds from a very early age. So the effect of this cochlear amplifier is it amplifies the sound being delivered to essentially the sensory cells of the cochlea by a factor of 100. It's responsible for the good frequency selectivity that we have, because it makes that resonant peak much, much narrower, and it produces these otoacoustic emissions, as they were then called, discovered by David Kemp. And the person who actually used this phrase cochlear amplifier, I believe, was Halliwell Davies, who was one of the key cochlear physiologists of the middle of the 20th century. And there were a number of things that all came together at the beginning of the 1980s, when I was fortunate enough to be involved for the first time in thinking about hearing. And one of the observations that was made was that it turns out that even from within the cochlea, you get the cells responding as though the basilar membrane was actually a very sharply tuned resonant structure. And this is an observation that was made by Ian Russell, who is sitting here in the audience. But the idea that the outer hair cells were responsible for this amplification was something that was a little bit slower in coming. So let me show you, first of all, what happens if you build a cochlea on physical principles alone.
They say some silly things.
So that's the stimulus that we're going to put in. And I'm going to now try and play this. I'm not doing very well here. Sorry about this. Here we go. And you can see here, what we have here is the basilar membrane, but idealized, so it's sort of sitting in a box. And down the bottom here, we have a sort of three-dimensional version of the basilar membrane, with the sound coming in here at the stapes end, and the different colors indicate the vibration amplitude. And you can see that as this sound, some say some silly things, is played through it, you get a kind of ripple traveling along the basilar membrane. But it's not a very well-formed ripple. It just looks like a sort of slow swell rather than any well-structured movement of a vibration pattern of the basilar membrane, which could give information to the brain. However, if you put the outer hair cells in, then you end up with, essentially, you can see a much more precise pattern of vibrations, as though every little portion of the basilar membrane is able to resonate free of this drag produced by the viscosity of the fluid. And you can see this on this pattern here. You've got these individual response regions of the basilar membrane, which is going to convey information up to the brain. So this is a model that's based on some software developed by colleagues of mine, Renato Nobili and Fabio Mamano. And this arose, in fact, out of a Royal Society soiree where a colleague, Matthew Holly, and I were demonstrating what we thought was happening in the ear, and they took our rather crude model that we had and pulled it all to pieces and then built the software that essentially tried to reconstruct what might be happening in a real cochlea. So let's now start turning to the physiology. We have here a basilar membrane which can vibrate, and the question is, where does this cochlear amplifier come from, and how, in fact, is it working? So all the way along the basilar membrane, if you take a cross-section through this cochlea, it turns out to have a structure which, in fact, doesn't contain just two compartments, it contains three compartments. This middle compartment is the compartment which contains the sensory cells, the hair cells, which are going to be involved in cochlear amplification and signaling off to the brain. And if you look a little bit closer at that structure down there, sitting on the basilar membrane, it's a long epithelium. This section here repeats itself 3,500 times along the basilar membrane. And this structure here, the organ of Corti, contains a variety of different cells which then are going to be essentially the physiological way in which the basilar membrane is amplified. So it contains cells which are known as the outer hair cells because they're sitting furthest away from where essentially the nerve enters, and the inner hair cells, which are the ones which are going to be connecting up to the auditory nerve. And the other thing you'll also notice about this diagram here is that these outer hair cells are sitting more or less over the middle of that basilar membrane partition all the way along the cochlea, so they're in a position to influence the movement of the basilar membrane. And that's what I'm going to be telling you about. Now let's look a little bit more closely at the hair cells themselves. This is now looking down onto the organ of Corti from above using an electron microscope, and you can see this very precise patterning of the hairs or stereocilia of the individual hair cells. You can't see the cell bodies because they're buried underneath this top sheet of this epithelium. There are other cells in here. In fact, there are many different cells in the organ of Corti, all with very, very specific names. But there are 3 rows of outer hair cells and 1 row of inner hair cells. Why are there 3 rows of outer hair cells? Well, again, what are the outer hair cells doing? And curiously enough, it wasn't immediately obvious, although from about the mid-70s onward, evidence was accumulating that outer hair cells might in fact have an interesting role. They would actually be not just passive receivers of sound, but they're doing something, motors. So let's have a look first of all about the way in which a signal could get into one of these hair cells. This is a picture that comes from a very nice review by Robert Fettyplace and Carol Hackney, and it shows the organ of Corti again. Here is the basilar membrane and the fibers that von Helmholtz was seeing run backwards and forwards like so. And over the top of this organ of Corti, or at least a part of it, is another membrane called the tectorial membrane. It's almost transparent, so it would have been very difficult for Helmholtz to see. And the idea is as the basilar membrane moves up and down, this tectorial membrane slides backwards and forwards and essentially moves the hairs of the hair cells backwards and forwards and essentially provides a mechanical stimulus to the cell body, to the hair cell, which is sitting as a part of these stereocilia. Now, the other interesting thing about this whole movement is that it's really small. It's just nanometers, molecular dimensions. So that's another technical reason why it's been very difficult to make precise in vivo measurements of the movement of the basilar membrane. But a number of labs have been very successful at doing this, and we're beginning to have a complete picture of what the underlying mechanics of the basilar membrane is. So let me briefly tell you about how mechanical movement is converted into an electrical signal in the hair cells. Here's the 3 rows of outer hair cells. Altogether, when you're born, you have about 15,000 hair cells, of which about 3,000 are inner hair cells and about 11 to 12,000 of these outer hair cells, stretching all the way along the basilar membrane. And right at the top of these stereocilia, the hairs, is the cluster of proteins which convert mechanical movement into an electrical signal. So let's have a little look at that. Here's a very elegant electron micrograph taken from above of the stereocilia of an outer hair cell. So the cell body is that wide, and in fact the cell body would extend down underneath the screen. But these stereocilia sit up as little stiff rods poking up from the top of the hair cell, and there's about somewhere between about 50 and 80 of these stereocilia per cell. So altogether the cochlea has getting on for close to a lot of individual stereocilia. Now, if you look very closely at one of these little regions here between the stereocilia, it turns out that they have what looks like a link that runs between one of the shorter stereocilia and the next one up. On this model here, that runs between this middle row and the next row of stereocilia, and this shorter row and the next row of stereocilia. And these were first, the presence of these links was drawn to the attention of the community, of the hearing research community, by Jim Pickles in the mid-1980s, and they were called tip links. And you can see that they have quite complicated structures. So they may fork at the top, there may be a little bulge there, but we believe that the actual machinery which converts movement into an electrical signal sits at the bottom end of these tip links. What does it look like? Well, it's like this. Here's the tip link. It's made out of a couple of proteins whose identity we now know, cadherin 23 and protocadherin 15. And sitting down the bottom here is the proteins which then allow an electrical current to flow into the cells and produce an electrical signal in these cells. And although we're getting closer, and despite a lot of work by many laboratories, we still don't really know how mechanotransduction in hair cells works. We don't know how this cluster of proteins, and there are many proteins, some of which are affected by genetic mutations, conspire together to form a very effective way in which essentially movement of the stereocilia can get converted into an electrical signal. Now, there is one interesting consequence, however, of this arrangement. You can see that it's, in some senses, it looks as though it's like a bit of machinery, and so therefore it ought to work very fast. But on the other hand, you could imagine that if I push these stereocilia backwards and forwards, if I pull them in that direction, then it's going to put a tension on this link and possibly do something to the cluster of proteins down the bottom there. But if I push them in the other direction, there's always a danger that this link might actually crinkle up in a little way. It'll go slack. And that means there's a difference between pushing the bundle in one direction compared to the other. And there's quite a nice demonstration of this by, that was really highlighted by Jim Hudspeth and his co-workers at the Rockefeller University. And so if I play to you, if I go back one, sorry. Can I go back? Thank you very much. If I comes to you trying to use a slightly different computer, my apologies. If I play this tone, I'm going to play two tones together, which is 1200 and 1000 hertz. Can I play that again, please? So what you can hear is those two tones together, and you would think that they ought to beat together with a difference between these, so that it should beat at 1200 minus 1000 or 200 hertz. In fact, because the movement is asymmetrical, there's much more of a mixing of these tones, and in fact, there's, as well as the 200 beat tone, there's actually another one which is composed of twice the 1000 hertz minus the 1200 hertz. In other words, it's a kind of phantom tone of 800 hertz. And you can convince yourself that there is actually this phantom tone, or combination tone as it's sometimes known as, but if you play at the same time a tone of 804 hertz, and so the 804 hertz tone will beat against that 800 hertz tone. So if we can play that one, I'm still pushing the wrong button, sorry about that. Do you hear the warble? So that's because there's this imaginary tone which you can hear. And it's not just because there's distortion, I hesitate to say, in the Royal Society's audio system. It's actually present in your ear. Your ear is not a completely hi-fi device. It has a bit of intrinsic distortion, and you can convince yourself that by even playing on a well-tuned piano. You can actually eventually work out that there is this phantom tone, this combination tone sitting in there. So let me now turn to the main topic of this lecture, which is what are the outer hair cells doing? I've hinted that the outer hair cells are the amplifier, and I've hinted that the inner hair cells are part of the sensing system, as it were, the microphone sending the information up to the brain. How do we convince ourselves that the outer hair cells are indeed amplifiers? Well, the evidence came in a rather surprising way from work from Bill Brownell in the mid-1980s, who was working with his, during a sabbatical year at Geneva. And he observed the fact that these hair cells change length when you change the potential inside them. Now, the techniques they were using at the time were good, but it really took the full power of what's called patch clamp electrophysiology with wide bandwidth to be able to show that these outer hair cells had most surprising properties. And let's see if I can show you some of this in a rather more vivid way. Now, I'm afraid to say that for copyright reasons, those of you out there in the webcast can't actually hear the sound, but what I'm going to show you is an outer hair cell which is being stimulated through a micropipette, a patch pipette, which is changing the potential inside the cell. So this is an outer hair cell which has been dissociated out from the organ of Corti. The hairs would be up the top here, the stereocilia are up the top, and we're patching it down the bottom there. Normally, of course, the movement of the stereocilia would be the thing that's generating the change in potential in the cell. But if I do that now, and if I can now run this, can you run this video for me? And I hope what you noticed was that as the cell got longer and shorter, it actually changed its width a little bit. It's a little bit like a sort of balloon, which if you squeeze it, it sort of gets longer, and if you can somehow shorten it, then it gets a little bit wider. And the most remarkable thing about this is that you can actually take a photograph of these cells. You don't have to take a video, and provided you make the inside negative, then the cell gets longer, or the inside positive, it gets shorter. And that movement is about 4% length, the change in length of the cell. So this is a photograph taken by my colleague Matthew Holley, who was interested in the possibility that the mechanism might in fact involve one of the conventional mechanisms of cell motility that we know about. In other words, this is a muscle. No, it couldn't be a muscle because muscles, you stimulate them and they tend to relax again. This is something strange about this one. You stimulate it by a change in potential and it stays that particular length. And so the question really was, what's the underlying mechanism that's responsible for making these hair cells change length in this particular way? So what we ended up finding out about these cells is that they change length when driven by the intracellular voltage, the voltage across their membrane. Every little bit of membrane turns out to respond by a change in area. So it uses what's equivalent to a change in area. And you can imagine that in the balloon case, because there's one way of actually making this balloon get shorter, which is to inflate it. I'm not going to try. It's quite hard enough to blow this balloon up as it is. But even if you try and inflate it, it turns out that most biological mechanisms we know about aren't quick enough to inflate or deflate a balloon very quickly. And this change in length of the cell happens very quickly. But what we believe is that it's actually a motor in the side of the cell which is actually causing, by changing the tension in the cell, to make the cell get longer or shorter. There are huge numbers of copies per cell, in the order of millions, and if you drive it really fast, but with suitable electrophysiological techniques, you can show that these cells move at speeds well over 50 kilohertz. In other words, quite fast enough to be involved in many of the acoustic phenomena that we know that the inner ear is capable of. The evidence that there are so many copies of these of this particular motor comes from a beautiful picture by Bashara Kashar at NIH, which if you enlarge, here's the outer hair cell with its stereocilia, here's the side of the cell. If you enlarge this little region by about considerably, you can see that it's actually densely packed with particles. And if a hair cell were the same height as an adult human, then each one of those particles would be about the size of a grain of sand. So you can imagine that you can get a lot of these particles into the side of a hair cell. The molecule responsible for that particle was eventually identified in 2000 by a group at Chicago under Peter Dalosh. And when you discover a new protein, you can usually, or even a gene, you can have the right of calling it something, and they called it prestin. It's because it moved the cells presto, or fast. And the other problem, as I'm sure you know, is that when you discover a sequence for a protein, there's no guarantee that you actually know what its structure is. So you have to resort to modeling techniques to try and work out what its structure is. It's clear that it was actually in the membrane. And when you remove this protein from the genome, essentially the animals become deaf. That's fine. But the actual structure of this protein remained far from clear. It was thought to have 12 transmembrane spanning regions sitting in the membrane of the cell. And then there's a cytoplasmic region which contained this strange thing called a Stas domain, which is a sulfite transporter anti-sigma domain, which people in the bacterial field knew all about. And the other surprising feature of this is that this protein turned out to be a member of a superfamily of membrane transport proteins. It wasn't even a motor protein as such. It wasn't a muscle protein, it wasn't a kinesin or anything like that. So it was member number A5 of the superfamily of solute carrier superfamily number 26. And these are proteins which move chloride and bicarbonate backwards and forwards across membranes in whole varieties of different tissues. You have chloride and bicarbonate movement in the red blood cells of your body, but that turns out to be a different family. This is a chloride or anion, negatively charged ion bicarbonate transporter that's found actually ubiquitously in a whole variety of other tissues. So the question really is, okay, we've got a motor, but is it actually a transport protein or is it a motor? Maybe the Dalosh people actually made a mistake. Unlikely, but it's still always possible. One should always doubt these things. So let me just provide you a little bit of evidence that these prestin motors really are transporters as well. So here is the organ of Corti sitting on the basilar membrane, and here's the cells we've been talking about, and here's the outer hair cells, and all the way down the side of it we have a prestin molecule. But it had been known for a few years before prestin was discovered that these cells were really good at regulating their intracellular acidity, or pH. And for that, you need clearly something which is a chloride or bicarbonate exchanger, because that's the way in which you regulate intracellular pH. And so we decided to look at that problem using imaging techniques. And the way in which you do it is to link a detector of acidity to the prestin molecule. So you stick a molecule called fluorine onto prestin, and that detects any pH changes near prestin. Fluorine is a modified green fluorescent protein, a modified GFP, which was originally developed by Gero Miesenblock, who is now in Oxford, but when he was working in James Rothman's lab in Yale. And when you express prestin fluorine in a kidney cell, not a hair cell, then it tends to find its way onto the membrane of the cell, exactly where the prestin would be in a hair cell. And as a control, what we did was to attach fluorine to something which wasn't prestin, but something would just stick to the membrane, to the inside surface of the membrane. And so the experiment is the following. If you expose a cell having expressed any of these proteins to carbon dioxide goes across membranes, the intracellular cytoplasm becomes more acid and the pH falls. If you expose it to bicarbonate goes in and it makes the inside more alkaline and therefore the pH rises. But if you have something which can exchange chloride and bicarbonate, then the question is, what would it do? We would hope that it might actually increase the rate at which the cell becomes more alkaline, because you can load it more quickly. And that appears to be the case. So this is the way in which you do the experiment. You monitor the pH with looking at the fluorine. In high chloride, where there's no driving force for bicarbonate, you expose it for bicarbonate, and it doesn't matter whether it's the prestin cell or whether it's the control cell, the cell begins to recover, but not particularly quickly. If, on the other hand, you now expose the cell to low chloride and give it bicarbonate. That produces a chloride gradient which will suck bicarbonate into the cell, so the cell recovers much more quickly and becomes alkaline much more quickly, and Preston is clearly transporting bicarbonate. You can do exactly the same experiment with chloride, except you don't use fluorine, you use yellow fluorescent protein. This is another modification of green fluorescent protein, which turns out to be chloride sensitive. And you can see that when you do that same experiment by producing the right conditions for Preston to really suck chloride out of the cell, then you get a pretty healthy chloride signal compared to the control. So that's where one sort of stands. And so the question then that works is, how could Preston even work as a molecular motor? What's a transporter doing behaving like a motor? And if you think about it, what a transporter is doing is it's taking in something from one side of the membrane, undergoing a flip, and then essentially kicking out that substance to the other side, and possibly flipping backwards and forwards. So it looks as though what may be happening is that Preston is behaving like a rather inefficient transporter, but it's just using a bit of the transporter cycle to flip through, as it were, to flip but not flop. So it's undergoing what looks like a change in area, and because these particles, these molecules are crowded so closely in the membrane of the cell, you end up with essentially the molecular crowding making the whole cell change length. All well and good, and it's become a little bit more, the situation's changed a little bit recently because we still don't really know what the structure of Preston is. There was some remarkable work done by Dominic Oliver and his group at Marburg, which said that they didn't quite believe the 12 transmembrane spanning model that was originally proposed. And they, based on the similarity of this SLC26 family to the uracil transporter family, they said it's probably much more likely that it's got 14 transmembrane spanning regions in it. In other words, it's a duplication of two 7 regions next to make a 14 transmembrane region, spanning region. And the idea was that the transporter looks like two sheets which flips backwards and forwards, and in the process of doing that will actually transport chloride and bicarbonate in opposite directions. Now, a couple of years ago, it turns out that even bacteria have a member of the SLC26 family, and the crystal structure of the bacterial SLC26 family is known. And that turns out to have 14 transmembrane spanning regions with this plate that the Oliver and co. had actually predicted, behaving as the transport pathway through which you can transport anions and bicarbonate. And then the Stas domain, which sits down the bottom here, is the one that appears to be also part of the structure, but we don't know how that's actually associated with the main bit that sits in the membrane. The other bit of information that we also know about Preston is that it's a bullet-shaped particle. The particles that you see in the membrane are too big just to be a single molecule of Preston. It looks as though there are probably multiple copies of Preston. And this study supported the idea that there were probably 4 of these Preston molecules joined together as a 4 subunit structure. In other words, it's a tetramer. And the most likely thing is a dimer of dimers. You end up with 2 molecules joining together and then two of them forming the tetramer. So here it is sitting in the membrane. Quite a lot of it is actually in the membrane, but there's also an awful lot of it sitting in the cytoplasm as well. Now, I'm going to indulge now in a little bit of sort of fantasy to show you how all these bits might come together. Imagine that Preston with these 2 plates is sitting in the membrane. Here is the 2 regions which are transporting chloride and bicarbonate, and here is the Stas domain sitting in the cytoplasmic region. So it's able to transport chloride out or bicarbonate in. And now imagine that you end up with 2 of these sitting next door to each other, and possibly the two of them join together, so you end up with essentially a 4 subunit structure. And you could sort of imagine that when you've got a structure like that, it just takes a small rotation of the molecule to actually give this 4% area change. A 4% area change is just the diameter of a hydrogen atom, not very much. So This, I have no particular reason for suggesting this, other than just to suggest that maybe that what's actually happened is that the evolutionary changes that have happened in Preston, in becoming the mammalian Preston, are evolutionary changes in the Stas domain, which have ensured that it acts like a plate against which a sort of rotator can then move and make this area motor change very, very quickly. Now, I'm well aware that amongst the regulations of the Croonian lecture, as John said, there's, as well as being supported by rent from a tavern, one of the regulations of the Croonian lecture, if I can find the appropriate document, is that they were framed to discourage vague and insubstantial theories. And I'm afraid I've just offered you a rather vague and insubstantial theory. So let me just summarize that by saying outer hair cells are mechanically sensitive cells. The outer hair cells, but not the inner hair cells, are motors, or the technical term is actually they're an actuator. They don't supply their own energy from energy sources. They're using the electrical field that's generated within the cochlea. And they use a motor called Prestin, which is a member of an SLC26 family. And the suggestion is that Prestin is also part of the regulation of pH in these cells as well. And the amazing thing is that all hair cells that we know about have a Prestin-like homologue. And what appears to happen is that sometime during evolution, the outer hair cells in the mammalian system hijacked this transporter and used it as a motor to essentially amplify the movement of the basilar membrane. So, what have I shown you? I've shown you that when sound comes into the basilar membrane, it makes the basilar membrane vibrate in a frequency-specific way, and that essentially links up to the tectorial membrane, which enables the stereocilia of the hair cells to be deflected backwards and forwards. And then the outer hair cells use the change in electrical potential to feed back onto the basilar membrane, because they're sitting in the right position for their length to move the basilar membrane in such a way as to cancel the viscosity and amplify the movement of the cells of the basilar membrane. So what happens when you begin to lose your outer hair cells? Well, of course, what you end up using is a hearing aid to amplify the movement of the basilar membrane. And that obviously becomes critical where you begin to lose, where the outer hair cells have been lost, and so you can amplify the movement of the basilar membrane, not quite as well as the outer hair cells, but well enough for an inner hair cell signal to be generated. What's the evidence that outer hair cells are lost with age? Well, this is a remarkable picture of an electron micrograph from some post-mortem material from an 85-year-old that was collected by Tony Wright and Andy Ford at the UCL Ear Institute. And you can see that there's not a single sign of any of those stereocilia that we were looking at in the outer rows of outer hair cells. There are missing outer hair cells, but the epithelium has closed up over the gaps where it's self-repaired, where the outer hair cells were. We haven't ended up with more outer hair cells. We've ended up essentially with a repaired epithelium. But there are some inner hair cells still remaining there. Now, you can do the same experiment in a mouse. So here's a young mouse, an 8-week-old mouse. Here's the 3 rows of outer hair cells, 1 row of inner hair cells. And when you look at a 2-year-old mouse, in other words, an octogenarian mouse, it's only got 1 outer hair cell there, stained by this blue label that labels up the outer hair cells. All the sort of the skeleton of the hair cells are there, but there's no cytoplasm or stereocilia at all. These little spots that people at the front may be able to see better than those at the back are the spots that correspond to the synapses between the inner hair cells and the nerve. And you can begin to see that some of the synapses have also changed in the old mouse, as well as, although the stereocilia are reasonably okay. So let me tell you finally a little bit about the nerve fibers. We have 3 rows of outer hair cells, 10, and then each inner hair cell is connected up between 10 and 20 between 10 and 20 synapses, which connect up to between 10 and 20 nerve fibers. Each inner hair cell has a number of preferential pathways up to the brain. And if you look at, again, post-mortem material, so this is work now that comes from Charlie Lieberman in Boston, you can see that if you look at the low-frequency region and stain up the nerve fibers, there's a pretty rich collection of nerve fibers innervating the inner hair cells. But if you look at the high-frequency region, then the fibers become much, much more sparse. So it's certainly clear that you begin to lose, with age, the high-frequency auditory nerve fibers, but not completely. And you can see that in this rather more complicated plot, which plots the number of nerve fibers per inner hair cell as a function of where they are along the cochlea. So when you're young, you've got anything up to 15 to 20 or so nerve fibers per inner hair cell, possibly more. Most of them are around about the 1 kilohertz point. But as you get progressively older, you begin to lose those fibers, but you don't lose them completely. So there's a certain amount of interest being generated in the possibility that maybe one can encourage those residual nerve fibers to do the job that they're meant to do. So what I've told you about is this cochlear machine, this amazing machine that delivers information up to the brain. We've looked at the motor structures of the outer hair cells. We've looked very briefly at some of the inner hair cells. And all of these are sending the information up to the brain. And I think I really should say at this point that there's all sorts of risk factors, as we originally sort of mentioned. I mean, it's not just loss of outer hair cells, but there's noise, genetics, and possibly drugs, which can affect this very delicate machine. But to be able to hear well, you need this machine to be in tip-top condition. There are ways of compensating for lack of tip-topness, but not perfect. Now, in summary then, perhaps let me just end with a little aphorism from a Greek philosopher, Archilochus, who said, poid oidolopex al eikonos en mega. The fox knows many things, but the hedgehog knows one big thing. Richard Feynman, obviously I reveal my physics roots here, because he was influential in making many of us physicists first before we became physiologists, said to, is reported to have said to Freeman Dyson that scientists are either hedgehogs or they're foxes. I'm afraid this has been a bit of a fox talk, but I hope you will realize that in trying to understand the ear, you actually need to bring all sorts of things together. You need to bring molecular biology, you need to bring physiology, electrophysiology, imaging, increasingly psychology, the way in which the brain is working, and audiology to be able to understand all these things effectively together. So I should just say that, of course, that what I've been telling you has been the work of many people. I've been very fortunate to have wonderful graduate students, wonderful undergraduate students to do projects with me, excellent postdocs who've gone on to great careers in their own right, and of course a huge group of people around in the hearing research community who've fed information in and shared their information very freely. And I've been lucky in as much, and I've been supported by the Medical Research Council and the Wellcome Trust, and also I'd particularly like to say in this post-Brexit era that of course many of the things I've been telling you about originated from a EuroHear FP7 project supported by the EU. But last of all, thank you very much for bearing with me and having the attention to stay with this rather long and protracted story. Thank you very much.
So we have time for some questions. Yes, right at the back first.
It seems like everything obviously to do with the ear and hearing is incredibly intricate and small, and I was wondering how things are progressing with being able to do any kind of transplants when you're kind of becoming, more hard at hearing.
I mean, people are thinking along these lines. The problem with the ear is that it's known that the development of the human ear occurs between weeks 24 and weeks 26 in utero. And it looks as though there's relatively little turnover of the tissue in the ear after that. And so it may not take too kindly to being tinkered with. Transplant, maybe. I mean, there are ways, of course, in which you can get directly at the nerve. And, of course, the whole industry of cochlear implants, which stimulates the nerve directly but bypasses the hair cells, is one way in which people are thinking about this. The whole issue, I suppose, of even thinking of transplants is one of accessibility. And as I mentioned right at the very beginning, it's the inaccessibility and of the inner ear machinery, which sort of makes it a really difficult target for thinking of even quite crude surgical interventions.
One just in about 2 rows in. Was there a question over there? Yes.
Hi, thanks for the talk, really lovely.
You should say who you are. Sorry, I missed the first line.
My name's Tommy Jantz, postdoc at UCL. You spoke a bit about how the hair cells transmit information to the brain, and that's obvious. But what is the feedback from the brain to the cochlea doing? Is there anything known about that?
Fascinating. Yes, well, people have worried a lot about that. There are descending pathways from the brain, both to the outer hair cells and also the inner hair cells. In fact, this is one of the ways in which I got into this area, because Ian Russell and I ended up talking a lot about what the efferent system was going to be doing on the outer hair cells. And it's clear that when you activate the descending pathway to the outer hair cells, it turns them off a little bit. In other words, it makes them, it reduces the feedback. Not hugely, but sufficiently, but we don't know how much it actually does so under physiological conditions. And the thinking at the moment is that the main function of the efferent system, at least of the outer hair cells, is to make sure that the balance between, the sensitivity balance between the two ears is about the same, so that when you try and localize where an object is in space, you don't get an abnormally large signal from one side. And maybe this is a dynamic process that's actually going on the whole time. The significance of the descending pathways to the inner hair cells is a bit more complicated, and that descending pathway tends to, the efferent system terminates on the afferent boutons of the inner hair cells. In other words, it looks as though it's modifying the flow of traffic coming down the nerve rather than influencing the inner hair cell directly. But it could even be that the effect of the efferents is different at one end of the cochlea from the other. We don't really know fully at the moment, but very interesting question.
Yes. There's a microphone here, to your right.
Thank you very much. Peter Michael, I'm a retired engineer. Somewhere along there, and great, terrific, I began to feel that I was hearing the explanation of tinnitus, where it seemed to me that the positive feedback mechanism, which generates the frequency response, sharp skirts, was overcompensating. And that the fact that you could hear tinnitus sometimes in an adult, you could hear emissions of sound, seemed to link to that piece of your lecture.
Great guess. I'm sorry to disappoint you, that it's probably not quite correct. And the reason for that is that we believe that tinnitus is actually of central origin, and that tinnitus is rather like a kind of phantom noise. I do agree with you that it has all the ingredients for looking at the feedback could almost be thought of as a generating a tinnitus, but there are all sorts of little bits of evidence that argue against it. One of the most powerful ones is that, we don't take aspirins anymore for headaches, but when people used to take aspirins for headaches and perhaps overdose themselves sometimes, you actually get a tinnitus. One of the surprising things about aspirins is that they turn off the effect of the outer hair cells. And so when you turn down the cochlear amplification, the brain endeavors to compensate, we believe, for that by essentially turning up the gain further up the pathway. So it's a bit like the interstation hiss that you get on a radio set. And the other way of talking about tinnitus is it's like a sort of phantom noise. In fact, you're sitting just in front of somebody who can tell you all about phantom pain, because there's been a great discussion about whether there's similarities between tinnitus and mechanisms which generate the as well, neuropathic pain of various sorts. Well, with aspirin it's reversible. Clearly, when you have a tinnitus, there are some cures, maybe. Not, I don't even want to use the word cure. I mean, people are actively trying to think about ways of reducing the effect of tinnitus. The most effective way so far seems to be to address the question of attention. You get people to pay less attention to it. No, there is no emission. In very few examples, maybe. Again, the person next door to you will be able to answer that question. That's David Kemp.
So, another one. That's it in the middle. Can you see? Thank you.
Hi, my name is Benjamin Graham. I'm a pharmaceutical scientist. I was wondering about you were saying that as people age, they lose the hairs on their outer hair cells. But looking at the electron micrographs, they seem to be, the cells themselves were still present. It was just the hairs themselves that were dissolving. I was wondering about like the mechanism and the whys of that.
Well, I don't know. I'm not really...
Can you induce that sort of damage?
Can you induce? Well, yes, you can certainly induce it experimentally with drugs, because that tends to kill off the hair cells. You can certainly damage the cells experimentally with noise, because the machinery is obviously very delicate. There's clearly a sort of carcass of the cell which gets left behind. But it is a manipulatable sort of system in that sense.
Right at the back, can you see the...
Hello, Heather Jones, molecular biologist. Could you comment on the effect of possibly nutrition, or let's say calorie restriction, on the disappearance or apoptosis or injury of these outer hair cells?
Gosh, I don't know. I mean, what happens if you ask jockeys, if they starve themselves, what their hearing is like? I really don't know. Interesting, very interesting proposition. I have no idea at all. I'm sorry. Great, this is an example of a wonderful interdisciplinary question. I hope you'll follow it through.
Yeah, this one here, please.
Good evening. I'm Jack Golem, a retired pharmacist. I'm actually deaf with high-pitched deafness myself, which is why I'm here. I'm quite fascinated about the theories concerning the effects of Preston and how they work. I would like to ask you, have you ever considered the possibility of Faraday's laws of electromagnetic motion in the process, when you have the thumb, the first finger and the second finger, and the first finger represents the field, the second finger represents the current, and the thumb represents the motion, and perhaps it's the confluence of that with hydrogen bonds because of the high concentration of polypeptides, that it might be creating a sort of electronic plasma and therefore generating that movement.
There's certainly enough energy in the electric field to be able to do that. The evidence against that proposal is that if you remove the anions from the cytoplasm, Preston doesn't work anymore. So it's not just a purely electrical phenomenon. It clearly does depend upon the movement of ions. Having said that, there is clearly when Preston changes conformation, there are movements of charge within the protein itself. And a little bit of that may be susceptible to the kind of transmembrane field that you're talking about.
Okay, we're going to leave it at that, and it just remains for me, since Jonathan received his medal and his prize at a dinner last autumn when the other Premier Awards were given, it remains for me to say thank you very much for a wonderful talk, just as good as we expected, and I have to say there is nobody of my age that doesn't always respond to Rock Around the Clock whenever they hear it. Thank you very much.
Thank you very much, John. Thanks, Dave.